Hello, students. Welcome back to my class. So, in the previous class, we have discussed about the layout of course, that is nucleic acids, and we have discussed about the various topics that we have to cover in this course. So, we have four topics basically in this particular course that we have to cover. Also, I told you the outcomes and the assessment method for this course. So, today here I am with you with my first topic, that is introduction to nucleic acids and its classification. So let's start. So nucleic acids. Nucleic acids, maybe some of you are knowing already. So they are basically naturally occurring biopolymers, which are capable of being broken into small molecules, which is phosphoric acid or sugar-based molecule or a mixture of organic bases. These organic bases basically they are purine-based base and pyrimidine-based base. So you can see here the structure of this purine. This is the structure of purine, and this is the structure of pyrimidine. So pyrimidine is a six-membered heterocyclic molecule which contains two nitrogen atoms at 1 3 position, and purine, it contains two cyclic rings fused with each other, and both the rings contain two atoms separately. If you look at this six-membered ring, this is look like the pyrimidine ring. It has two nitrogens at in 1 3 correlation, and this is the another ring that we call imidazole ring, which also contains two nitrogen atoms. So these organic bases are basically their derivatives. These are not those bases, but they are derivatives. Okay. Talking about more little bit for this nucleic acid. So they are long chain like molecules. You know that since they are the polymers, so polymers, they can be long chain. They have various classifications of polymers. So they are like long chain time molecules, and they contains nearly similar type of units that we call nucleotide. And if a polymer is contains similar type molecule, repeated units of similar type of molecule, that molecule that is known as monomer of that polymer. So we can say that nucleotides basically are the monomers of these naturally occurring polymers. And this nucleotide basically contains these three types of molecules, that is phosphoric acid fragment, sugar, and a mixture of organic bases. Okay. Now these nucleic acids basically they have the ability, they serve as the primary information carrying molecules in the cell, and they play a very special role in directing the protein synthesis. Now moving further, so the sugars that we can obtain from these nucleic acids, or the sugars which are available present in nucleic acids, they can be two sugars. There are five carbon-based sugars. One is the beta D ribose, another is the beta D 2 deoxyribose. If you look closely to these two structures, you will see a difference at carbon number two. In this beta D ribose, at carbon number 2, we have hydroxy group downwards, whereas in this beta D deoxyribose, we don't have any hydroxy group at 2 dash carbon. There are just two hydrogens. Okay, so this is a very small difference between these two, but they make huge difference in the structure of nucleic acids. Now, this is the phosphate group, which we will get after the cleavage of this nucleic acid. Now, these are the two purine-based bases. One is the adenine, another is the guanine. So these are two bases which contains purine system. And here we have three pyrimidine-based base. One is the thymine, as is cytosine, and one is your, you can say, just a second, I will try to. Sorry. Third one is the uracil. If you look closely to these three structures, they are the derivatives of this pyrimidine system, and there is very minute difference among these three structures. Uh, if you look closely, thymine and cytosine, this is the only difference at this carbon. Cytosine here has hydrogen, whereas thymine at this position it contains a methyl group. And in uracil, this you can say this NH group is replaced by the C double bond group. Okay, and also here this is C double bond O, and uh, other rather than being a next group as we have in cytosine. So you see, we can see here that thymine is a derivative of uracil. Okay, so if you know either one of the structure of these three, you can just remember the other one. So here I have two questions for you that you will see in your screen. You have to answer of these two. Okay. Now, coming to the classifications of these nucleic acids. So, if we want to classify the nucleic acids, so broadly we can classify into two types of nucleic acids. One is the ribonucleic acid that we call RNA. Another is the that is deoxyribonucleic acid, that is DNA. Now here you can see this fragment of RNA. This is just a portion of RNA. It contains, you can see here, the four bases: adenine, guanine, cytosine, and uracil, and this sugar, okay, ribose sugar, and the phosphate groups, which are linked each other through a phospholinkage. The systems adenine, the sugar, is linked with the another sugar or base system through this phosphate linkage. Now, RNA can be further classified as messenger RNA, transfer RNA, ribosomal RNA. These are some other types of RNAs that we can have. Now, likewise, if we look at DNA structure, this is the fragment of DNA. Okay, this is just one fragment. It also contains the four bases. One is the guanine, thymine, adenine, cytosine. Okay, and this contains a sugar. Okay, and these sugar and guanine fragments, they are joined with each other through a phosphate. Okay, so these are just simple two types of nucleic acids. We'll discuss later on about the structure of these RNA and DNA in detail in upcoming lectures. Now, the difference between RNA and the DNA. Okay, so we can see the difference between RNA and DNA. There is very can say minute difference between these two systems. uh, just a second. Okay, there is very minute difference between these two molecules. In RNA, the sugar system that we have is a ribose sugar. In DNA, we have a deoxyribose. The other difference is that the primitive based base which are present in RNA, they are cytosine and uracil, whereas in DNA these are cytosine and thymine. There is a very minor difference between the two bases. Okay. Now RNA exists as a single strand, whereas DNA exists as a double strand. Okay. We'll discuss later on about the single strand and double strand, how and their structure, how they look like. Okay. Moreover, the backbone of the nucleic acid consists of alternating phosphates and pentoses. That means the phosphate group and the pentose, that is sugars, are alternative to each other, and the bases, that is purine or purine based bases, they are attached to these sugars in their backbone. So this is all about just basic, to give you basic idea about the nucleic acids. I hope you have understood, and maybe you will be able to learn more about the structure of RNA and DNA in upcoming classes. Okay, so thank you so much for watching.